here we go. All right, so for starters, we looked yesterday at prokaryotic gene regulation, and the general trend is this: a promoter with a whole bunch of genes downstream from it means that all you need to do is get transcription going once, and the whole process is going to go all together. So all those genes that are related in terms of function, they're all going and they're all happening at the same time. Um, so, Juliana, no, I'll let you know when you should jot anything down. But things are a lot different in eukaryotes. Namely, we don't have operons. We don't have one promoter in charge of multiple genes. Instead, what we have are promoters, and the promoters can be the same, but they're going to be in different locations. And this is how we can coordinate control of gene expression. And what I mean by that is if this was a prokaryote and the orange, green, and pink genes all needed to be transcribed at the same time, then one single promoter would have all three of them. For us to be able to coordinate our control, we've got to activate promoters at different locations in the genome. And in fact, I should have written this term down, so I'm going to write it in the chat. But coordinated control is going to refer to our genome being able to turn on similar genes at the same time if they need to be turned on. So this is how we express similar genes, whereas in prokaryotes, they're doing it using an operon. So if you do hear that term coordinated control, like I wouldn't expect you to spew it back out, but as long as you do understand that that just means that we're taking genes in different locations and synchronizing when they are being expressed, okay? And what's really cool is even though they're each going to have their own promoter, their promoters can be the same. And if the promoters are the same, then that's going to mean that they respond to the same transcription factors. Remember that transcription factors can either increase or decrease whether transcription happens, but in eukaryotes, you absolutely need them in order to be able to have the RNA polymerase bind. So if you've got a whole bunch of promoters that are exactly the same in different areas of the genome, and you get the nucleus is awash in transcription factors of a particular type, then you're going to, at the same time, all of them are going to have RNA polymerase bind, and they're all going to be transcribed at roughly the same time. That's going to allow you to make the different mRNAs that then will go into making the proteins during translation. Is that making some sense so far? Okay. All right, so I have a little bit of a video that we can watch that's going to explain why this is so important. And this is the part where I say, I don't know, because just in general, it is incredibly difficult to study what we are going to look at next. I just already said that. Transcription factors are going to be at different parts in the genome. Ooh, and one thing that's really cool in terms of research and what biologists are trying to figure out is exactly how all the chromatin fits together. And why is it that like some of it really loops way off into other areas of chromatin that maybe belong to a different chromosome? Anyway, so this is just a computer model of one of the hypotheses of how the chromosomes may arrange. All right, so here is something that my guess is that you have wondered before. Maybe, maybe not. Maybe I'm just weird. I know I'm just weird. Has anyone ever wondered how it is that you can start as a single cell, which, spoiler alert, you all did. 2 cells fused into one, and that was the first diploid cell that would eventually become little baby Annie or little baby Aaron. Sorry, you two are just the first 2 that are on my screen, right? How is it that that one cell divided to make You keep in mind that every cell in your body, very few exceptions, have exactly the same DNA. Okay, so as we're going through, I'll kind of narrate, but I'll try to not narrate too much. Let's skip to where there's, okay. So first thing that's going to happen is there's just a whole bunch of cell division, just general mitosis that's putting more and more cells into this little ball, this little cluster of cells known as the zygote, and then turning into this ball of cells kind of with an empty interior that becomes what's called a blastula. You don't need to know this. This is not anything that I'm going to test you on. But what's really interesting is even by, what is this, day 6 after fertilization, you see how they're distinguished by different colors? Now, of course, an actual embryo that is forming isn't going to be different colors, but they will be different cell types. Even this early, what we see is that some of the cells right here in blue and in yellow have migrated in one area, and then you've got this outer perimeter that are in kind of that maroon. They're different already. They have different gene expression. They call this hatching. It's more like you break open a hard shell, which I guess technically is hatching, but anyway, side note. All right, so all of that happens before someone even knows that they're pregnant, because guess what? Nothing has intruded into their body yet. But at this point, that little tiny embryo is digging into the sidewall of the uterus and then starting to communicate. And now these cells are each differentiating. They're each becoming a little bit more specialized and more specialized. And on the inside, that inner cluster of cells makes a flat cluster of cells. This part is cool. So this is what will become the baby. And if we take kind of a cross section, even under 3 weeks old, it's already starting to form a body plan with inside tissues and outside tissues, and the beginning of a neural crest, like a spinal cord. So cool. This thing has only been growing for 3 days. It's already got kind of or 3 days, 3 weeks, like a brain type area. You guys used to look like this. Weren't you so cute? You can probably start to tell what's going to be what here, although it doesn't even have leg buds coming out. And yes, you used to have tails. Aw. Some of you still might, I don't know. That's kind of your business, I guess. So has anyone ever wondered about that or thought about that in any kind of detail? How is it that the cells know what to do? Does anyone find that interesting at all? Meh? Or are you just like, they just do it because of magic? All right, hold on a sec. So here's what I'm going to do. I'm going to go into detail because when it comes to eukaryotic gene control, deciding when genes do or don't get expressed, a huge amount of this comes down to understanding cell specialization. Okay, so I'm going to go into some detail, none of which you have to write down until I tell you, But here's the big picture. When we first start with a fertilized egg, that's called the zygote, and the first thing that it is going to do is just divide. The DNA that is in its nucleus just goes through mitosis. Nothing unique here, nothing crazy. Just a simple process of mitosis to make a 2-cell structure, a 4, an 8, and a 16 cell. They just call them that, right? 16 cell stage. And every single one of these cells is completely identical to each other in terms of their genetic material. And all of this DNA is what we call expressible or on. Have you ever heard about turning genes on and off? So in this very, very early cluster of cells, all of the genes are on. All right. As this cell, or as this little ball of cells continues to divide, it's going to make a little empty cavity in the middle, and then cells are going to start to migrate to form this little cluster. You guys saw that in the video. But what's really important to understand is that these cells are now inherently different than the ones around them. The ones in the middle are what are called the inner cell mass, and the ones that surround that are called the outer cells or the trophoblastic cells. What that really means is these cells here are going to be destined to become the fetus, all the structures that are human. The trophoblast is going to be all the structures that are not going to be part of the human. They're just part of the supporting structures, the placenta, the amnion, all of those other pieces. So let's go back a second here. We have a cluster of cells. It has not even implanted into the mother's uterus yet. It's just floating along, bouncing down the fallopian tubes, and it already has a basic plan established. Some of these cells are going to be part of the placenta, and some of them are going to be part of the baby, which means something has to now be different, but they still have exactly the same DNA. They have the exact same genome, all the same sequences of bases, all 3-point-some odd billion base pairs, okay? And so something has to have happened here. So let's take a look at the differences in the DNA now at this point. We talked about already that what we can have is different areas of the genome with the same promoter. So they'll have the same response to certain transcription factors. So far, so good? And so it logically follows that if some have one promoter, then we could expect that others will have a different promoter. Now, I don't know how many different types of promoters and combinations of transcription factors, but what's important here is that if you are a gene that is controlled by a green promoter, then you're not going to necessarily be expressed at the same time as if you were a gene controlled by a pink promoter. That means that if I've got transcription factors that bind to the green, then those are the ones that are going to be expressed. If I have transcription factors that bind to the pink, then those are going to be the ones that are going to be expressed. Are we still so far so good-ish? Because now we're getting into something really important, which are called homeotic genes. They're genes that control the transcription factors, because guess what? These are usually proteins. So somewhere in the genome is the gene that makes one of the earliest transcription factors that are going to then activate a whole bunch more genes. Again, more detail than what you really need to worry about here. But the big idea is that if we can go all the way back to the earliest steps, we can start, in terms of writing stuff down, you guys are good. Don't worry about it yet. Okay, so in terms of being able to kind of go all the way back to the very first genes that are active, if we can somehow turn off all genes having to do with becoming part of the placenta, for example, or all genes having to do with being part of the human, being part of the actual baby, then we can start to specialize those cells. So in the case of the inner cell mass versus the trophoblast, right? The ones on the inside versus the ones that surround them, what's going to happen with these guys that are in the middle is they've got to turn off all the genes that would cause it to become a trophoblast and vice versa. Okay. So this is where you're going to want to start writing some things down. We're getting into what is called epigenetics. And so when we talk about genetics, foundationally, we're talking about the sequence of DNA nucleotides, A's and T's and C's and G's. But when we talk about epigenetics, we're talking about changes to DNA that no longer includes the sequence. Okay. It's like a genetic code on top of a genetic code. And so when I say it's a little confusing, that doesn't even begin to scratch the surface of it. But what scientists have been figuring out in about the last 30 years or so are just all of the, no, a tiny, tiny fraction of some of the ways that genes can be modified without the DNA being touched. Okay. And so a few things that we do know is that these are usually temporary changes. They're not going to be passed down over millennia. But when I say temporary, it could be over one organism's lifetime. Some epigenetic trait markers can even be passed from parent to offspring. So for example, we know that if mothers smoke, even before they're pregnant, that their epigenetics change and it could affect the fetus, even though there's not a fetus at the same time that she is smoking. Mind blown. Okay. So when I say it's temporary, just keep in mind it can be heritable, but not over a lot of generations. But it can be passed from parent to daughter cell. So epigenetic modifications can be passed from parent to daughter cell in what we'll call kind of like a cell lineage. And ultimately, what epigenetics do are they control whether or not genes are going to be expressed. When you hear that genes are turned on or turned off, what we're really talking about here is epigenetics. And so there's going to be two types we look at. They're called DNA methylation and histone modification. And these are not super fancy names, given that DNA methylation means exactly that, and histone modification means exactly that. So what I'll do is kind of go over the two different types. We'll relate it back to the cells that we've been looking at and then continue to fast forward. Give me a thumbs up if you're good and all caught up to where we are. Okay. Just didn't want to stay for too long on this. And Julia, FYI, this is being recorded, so you can go back in and watch the beginning that you missed, but you'll want to take notes on some of this, okay? All right, so let's take a look at DNA. This is just any stretch of DNA. It can be the promoter, it can be, you know, a lot of different sections, introns, exons, but what we know about DNA is that it can directly be methylated, and methylation just means the addition of a CH3. If you don't remember that from Unit 1, that is one of your functional groups, you may want to jot down next to DNA methylation, in parentheses, CH3. So we're adding these little methyl groups. And if DNA is highly methylated, then it is not expressed. It's almost like a warning tape, like do not cross. Okay. Now, in addition, keep in mind that DNA is not this free-floating molecule. It is super duper condensed. And the first step in being able to condense the DNA, to coil it up, is to wrap it around the histone proteins. And each set of histone proteins is going to have DNA wrap around it twice. And by having it wrap, you make what is called a nucleosome right here, which is the DNA with the histones. Okay. These histones can all start attracting to each other and really super duper tightly coiling. And that's going to make the DNA inaccessible. If it's really, really super tightly coiled, the RNA polymerase cannot get to it. So what we see is that just like with DNA itself, we can methylate the histones. Actually, the histone tails, these little pieces come out. They can be methylated, acetylated. You do not need to memorize these all, which is great. What you should know is that by modifying the histone, you can either increase how likely it is to be transcribed or decrease how likely it is to be transcribed. In other words, an on or an off. We don't have operators. We don't have operons. Instead, we change which histones are modified so that we can turn genes on or off. So typically what we see is that cells who have certain genes turned off, when they divide, their daughter cells are also going to have those exact same genes turned off. So even though they have to remove the histones to be able to have DNA replication occur, once they rewind around the histones and recoil, the same patterns of histone modification are in the daughter cells. Mind-blowing. I love this stuff. All right, so let's talk about it in context. Why does it matter, right? We started looking at these guys right here that are in that inner cell mass. Well, importantly, some of the genes that they may turn off or turn on may be things like cell surface receptors. What do you know about cell surface receptors? Do all cells have the same receptors? Okay. So now if this cell here has blue receptors and this cell here has black ones, are they going to respond to the same ligands that are in their environment? No. Which means that even though these cells could be right next to each other within this little ball of cells, because some of them have had certain genes turned on that'll give them one set of ligands versus another, now they're going to continue diverging and going into separate paths. So we call this differentiation. So the cells are becoming differentiated to become specialized. Okay, so some information about differentiation and cell specialization. What that means is that we're sequentially turning off certain genes. And so by turning these genes off and turning different sets of genes off, that means that each cell is going to make and respond to different transcription factors. That each cell is going to have unique surface proteins, unique signaling molecules. Those proteins that are involved in signal transduction are going to differ. And so that means that even cells that are pretty similar, these ever so slight differences that do exist in what genes are turned off is going to make the difference between perhaps responding to a particular ligand in one way versus a slightly different way, or responding to one versus not responding to it at all. So you can see that this kind of snowballs and is going to continue making cells that are unique from one another. Ah, shoot, that's what I was afraid of. I didn't think I had another point to make, but then I. So I want you guys to think back to cell signaling. In cell signaling, you have a receptor, a signal transduction, and a response. And that is going to be for any kind of ligand that is polar, that can't get through the membrane. So remember that polar substances are not moving through the phospholipid bilayer. And so you're going to have a receptor that can bind to the ligand. We all remember this, or at least I'm saying terms that are familiar. Okay, so the ligand binds to the receptor. All the different signal transduction molecules that are on the inside of the cell will eventually lead to a response. And guess what one of the responses is? In fact, one of the most common ones is either increasing or stopping transcription. So that means that somewhere in there, transcription factors were either activated or suppressed. That's really cool. What's even more cool is for polar ligands, or sorry, non-polar ligands, like the steroid hormones, testosterone, estrogen, progesterone, so all the different sex hormones, those are able to get directly into the cell and they bind to an intracellular receptor. Well, guess what they act as soon as they bind? As soon as estrogen binds to an estrogen receptor, that complex becomes a transcription factor. That when it diffuses into the nucleus, it's going to go right on causing transcription to occur. So we are regulating gene expression by having different ligands and having each cell be able to respond to different ligands. Now, here's where the mind-blowing piece comes in, because we know next to nothing. But here's a pretty good diagram of what we do know. We do know that the earliest cells are going to be right up here, right? We go into the 4, 8, 16, et cetera, cell stages, and then they become more specialized, and they make that flat disc of different cells. And we know that the inside ones become the endoderm, which becomes certain tissues. And we know that there's the mesoderm, and they become different tissues. I started this by asking, what do you want to be when you grow up? And what are you doing now to prepare for it? So here's what we're going to think of. Up here, these cells right here. These cells can divide and become anything. And in certain ways, that reminds me of a little kindergartner. They have no idea what's in front of them. All the awful schooling, I mean, all of the opportunities that will fall into their beautiful, cute little laps. And as they go through, they begin to find interests. For you guys, at some point, science must have been like a thing, something sparked. And so here you guys are. Now, if we want to think about cells on this pathway, we can liken it to education. Is it possible for someone who has never taken anything beyond algebra to then decide, I'm going to be a math major and become an engineer? Maybe, but not super duper likely. So you have to start making certain decisions to get to an end result. The difference here is that you guys kind of have an end result in mind and you're working towards it, whereas what cells are doing as they're differentiating is just being influenced by what is around them, almost kind of being guided which way to go. It's the parent that says, you have to take more math and science, honey. All right, now that you're in school, you really need to take all those pre-med courses. And they're pushing you towards being a doctor. Thanks, mom and dad. No, just kidding. And so what we see are something called stem cells. You've heard of stem cells, yes? Take a second and tell me what you know about them in the chat. So what is a stem cell? What do you know? Or what do you associate it with? What are some things that maybe like you've heard about them? Okay. So when we talk about stem cells, what we mean are cells that have the capability to further differentiate, but they're not really functional yet. Are you guys potentially going to become wonderful pediatricians and nurses and veterinarians and all of those wonderful things one day? Yes. And are you on the path to that? Yes. But if I went and put you in surgery today, first of all, I'd be in big trouble. And you would probably also be in big trouble and things would not go very well. You're not there yet. You are like a stem cell. And stem cells are kind of divided based on how many different cell types they could still be. So what we see is a totipotent stem cell is one that can literally become anything in that organism, whether or not that's the actual organism itself or maybe the placenta. Absolutely no genes are turned off. And this is only going to be that zygote, those first cluster of cells. From there, it does get a little bit more difficult to distinguish between the different types of stem cells. So totipotent cells can be anything. Pluripotent are still able to be a lot of things. Multipotent are much more differentiated, much more specialized, kind of towards the end of the road. Okay. And the opposite of a stem cell then would be a mature cell, which now is only expressing the genes that it needs. Everything else is turned off. The epigenetics has, each successive cell division has turned off more and more genes until finally it is at the point that it is a functioning cell. And usually functioning cells don't divide anymore, or they have very limited division. These mature cells are usually in the G0 phase. Okay. So it's really hard to study this all in embryos because the moral piece of it is going to be a big piece, like the ethical decisions that go behind that. So a lot of research into stem cells come from adult stem cells. So yes, you do still have stem cells that are multipotent in your body. You could even have some that are a little bit further back in terms of programming, maybe what we would call pluripotent, but you should not have any totipotent cells in your body. If you do, that is potentially an issue. But multipotent cells, one of the most studied is what is called the hematopoietic stem cell. And this is the cell that has the ability to become every other type of blood cell. So has anyone heard of what happens if you can't make certain blood cells? What do you tend to get? For example, if you have the inability to make white blood cells because of cancer, that would be oh, I just saw, Monica, you have it. What is it?
Bone marrow transplant.
Hey, you've all heard of a bone marrow transplant, yes? So a bone marrow transplant is exactly that. It's taking the tissue from the bone marrow. And in that bone marrow are a ton of hematopoietic stem cells, which have the ability now to become a lot of different things. So totipotent means anything. This hematopoietic stem cell is not going to become a neuron. It is not going to become a muscle cell. It is too far advanced in its education that it's not going there. But we could argue about whether it's pluripotent or multipotent. Pluripotent, again, meaning it's got a wider range of what it could become. And it used to be that we just thought there were really kind of 3 stages of it can become anything. Now it's less specialized. Now it's more specialized. We really blur the lines the more we learn about this. But I would say hematopoietic stem cells are multipotent. They can be a lot of different things, but not everything. And so what they can become are any of this group here, which include the red and most of the white blood cells, or they could be the lymphocytes, which include your B and T cells that are involved in immune function. Now, what's the difference? If you've got a hematopoietic stem cell, it's going to continue to divide and make a ton more stem cells. But if there's interleukin 17, IL-17, that is a cytokine or a cell signaling molecule that is going to bind to the hematopoietic stem cell and change its gene expression so that now it's going to shut off all the genes. It's going to either methylate the DNA or modify the histone so that it can no longer become a red blood cell, a platelet, or any of these other white blood cell types. It shuts that down and says, you've got 3 options for your future. Okay. This is congratulations. You just finished your English degree and you're out of money, so you can't go back to college. And now you're really nearing the end of your education and you have only a limited number of choices of what path you're going to take moving forward, right? If you finish with an English degree and decide it's time to be a lawyer, it's not happening. You have to get more school. If you finish with a biology degree and you decide that you're going to be a history teacher, well, too bad. It's not happening. Luckily, humans are not stem cells. We can be reprogrammed. But anyway, so at that point, this cell can become even more specialized until finally, once it is a T cell or a B cell, it is most of the way developed. It is now a functional cell. It is actually doing something for the body. The question becomes, which cell types are expressing which genes and how did they get there? And once we understand how they got there, how do we reverse it? For example, when I mentioned stem cells, some of you wrote about injury repair or replacing damaged tissues. And so the biggest promise that has been discussed about what can stem cells do is this idea that one day we could have truly specialized medicine. So I'm going to give you 2 examples. First example would be you have a person who has a severe spinal cord injury and is now either para or quadriplegic. If you can repair that spinal cord, the potential exists for them to regain function of their limbs. The problem is that adults don't have very many of the stem cells that eventually become neurons. So where are those guys? Here, the neural stem cells, we have almost none of. They're in very, very, very short supply, which means we're not making more neurons. We're not making more nerves to replace that damaged tissue. Once it is severed, once it is damaged, that's usually the end of it. The question becomes, can we get more neural stem cells? And we don't have any of these stem cells that are along this pathway to be able to do that. But what if we understood stem cells enough that we could reverse the path, that we could get back to being a kindergartner or a, eighth grader, go all the way back and take a new route in education. In this case, turn all the genes back on and then just selectively change it so that it can become a stem cell for the nerves. Have you ever heard of anything like that? It hasn't been done yet, but we're getting there. The other option is, let's say that I have a failing kidney and, oh crud, there goes the other one too. And now I need a kidney. And you guys are also wonderful, and you are the first to line up for a kidney transplant for me. Thank you. I love you all. That's so sweet. But odds are I wouldn't be able to take your kidney because, well, your markers, your cells, and the proteins that your kidney cells have on them are not exactly the same as mine. And so my body would reject it. My body would be like, that's an invader, time to kill, which is not good. One potential for stem cells is to be able to take cells that are in one part of my body. All right, roll up my sleeve. Let's take a little bit of skin right here. That skin cell can then be reverse programmed and then go down the track of making an entire new kidney in a petri dish that could then be reimplanted into me. And I won't reject it because it's my cells, it's my DNA. They're making all the same proteins that my original kidneys made, but only this time it's working. A lot of possibilities are there, but it really comes down to understanding how are each of these cells influenced by the cells that are around them, and how does that determine which genes they have on or which they don't. So I'm going to stop the share real quick and go ahead and stop recording.